So I thought, I mean, there is nothing much for me to cover, but what I thought we will do today is perhaps go over the topic, and if anybody has questions. I think most people have submitted. There are a few. I think Varsha, you have a few that you emailed saying that past deadline. Yes, email them to me. So once I have all, I will go over them.
Okay, I'll send the mail to you today.
Yeah. Do send the mail. Do send them through email.
Okay.
Yeah. Yes. So I will start by taking any questions that if students have, then we can take it forward from there on. Or if there are, or else I will just go over our past slides and see if there are things that need a little bit more better clarification. So yeah, anybody with questions, comments, or any suggestion? Nobody? I taught really well, huh? So any idea on what will happen after the embryogenesis is over? Because we covered until embryogenesis. So after that, we have the larval stages. Anybody on takes on that? How does that develop? Okay, so I haven't shared these slides, but what I will do is that I will talk about the system that I work on, because I just want to give you um, a flavor of what exactly, at least take the blood tissue and explain essentially um, how during once you have the development of the embryo established, what is the mechanism by which the system then continues to develop, because there will be yeah, so I will just share these slides with you. Yeah, just a minute. I think everybody should be able to. I will share. I will email these slides. I mean, I'll put them on Moodle. I'll give it to Varsha. She'll put it up on Moodle. Okay. So does everybody see this? The late gastrulation segmentation slide?
Yes, Tina.
Yes.
Okay, great. Okay. Yeah. Yeah, so we have covered essentially. Now, before I begin, can any one of you summarize like the key aspects that lead to, so the key underlying principles that you feel, that you think are going to be universal to the establishment of self-fate or to the establishment of segmentation? So what are the kind of what is the kind of approach that the animal is utilizing to develop either itself as a whole in the context of developing these segments, or any way you see, or even if it is about achieving gastrulation or movements, what do you think is the animal constantly utilizing? What are the approaches that the process of development utilizes? Can anybody tell me, I mean, summarize or just tell me a few points, and we'll see how far the points address all of the aspects that we have covered. So what kind of approach, mechanistic approach, does the animal take in? Is my question clear to everyone?
Yes.
Okay, so
Not snake. I mean, I was going to say that one of the approaches they take is that they follow this sort of coarse to fine grain specification of fates. So first
sorry,
they
sorry, sorry,
establish
take what,
boards.
take what? I missed it. I think the net is a little bad. So what did you say? Can you please repeat?
Sure. I was saying that one of the approaches they say that they employ is sort of going from a coarse grained specification to a fine grained. So they first establish just broad anterior, posterior, dorsoventral, lateral, and all of that. And then as development continues, the information is more and more fine tuned to being
Granular.
segment specific.
Right. So my question is,
But
yes,
I don't know if that's what you meant by approach.
yeah, but no, that is what you're seeing is happening, right?
Yes.
Now, my question is, how do you think, what are the basic approaches, as in mechanistic approaches, that the animal utilizes, or this is the process of development, what you just beautifully described essentially is the process of development, right? So how is development achieving this? So it is doing the same thing time and again to do this job that you described, either for the animal as a whole or even in a specific tissue we will see in a bit. But what are the things that you see the animal is doing again and again?
By employing gradient of morphogens.
Super. So morphogenetic gradients, cool, nice, yeah. And then so morphogenetic gradients, but before, not before or after, but apart from that. So morphogenetic gradients will distribute the information flow in different ways, but how it is read, so post morphogen gradients, then what is the next thing that you see that the animal that happens again and again? Be it for, say, the expression of genes, how is it controlling?
Like presence of various transcription factors that
So that's good. That's absolutely the best starting point, transcription factors. And is there anything that catches your attention by seeing how the transcription factors work? So it is the morphogen gradients lead to transcription factors, but not that all of them come up at the same time, right?
yes.
Yes, so there's a spatial and temporal sort of segregation. For instance, I know that all of these early patterning genes, the pair gap genes, pair rule genes, segment polarity genes, while they are establishing the axes in the embryo, they're also established, for instance, across the neurectoderm. And so depending on where stem cells arise, they have all of this kind of positional identity. So that specifies how each neural stem cell becomes unique.
Correct.
Then there's also
I'm not no, I'm not asking what transcription factors will do, but do you see the way transcription factors are aligned in, like you said, temporal, correct?
yes.
Yes. So not everything happens at the same time, and that there is a hierarchical organization of the transcription factors.
Yes.
Right. So that will that is one other important aspect, that there are transcriptional factors and they are temporally organized, not just temporally organized by being expressed early and late, but there is also a hierarchy. And this hierarchical organization is going to be key in, say, stem cells and cell fate. So we talk about OCT4, NANOG, SOX, KLF. They come up first, and then there are key transcription factors that come up first. Then the relay process starts off activating the different set of genes again and again. This creates, this leads to, a developmental pattern where like you can take the example of stem cells itself, a pluripotent, and then the layers of potency keep changing through this transcriptional hierarchy. Yeah, good. So great. So then anything else? Morphogenetic gradients, transcriptional factors, their hierarchical organization, and anything else?
There is also a switch between, continuously switching between the enhancers.
Enhancer switching. Yes, multiple enhancers will be employed to, that's very nice. Multiple enhancers will be utilized for regulating. So it's actually regulating gene expression.
Yeah.
So
Yeah, there is also continuous change in the cell shape.
Yeah. So cell shape changes. Cell shape changes to bring about morphogenetic movements, and yeah. So so before we go into cell shape, but enhancer switching, I mean, this is under the broad realm of regulating gene expression. What else do you think is, so in this broad topic of regulating gene expression, what is the other thing that catches your attention?
Different concentration of mRNAs localized at different regions of the embryo.
So you mean RNA localization and
Yes.
the localized localized
Yes.
control of gene expression, right?
Yeah.
Be it
Yeah.
translation or just positioning of the mRNA.
Yes.
Yes. Good. And have we finished everything with gene expression? Is there anything else that catches your attention?
Receptors?
Receptors, can you please elaborate, Akshaya, or who was this?
Yes, Akshay here. So I
Yeah.
was thinking, so although say the periuterine fluid, for instance, it provides various ligands.
Speak a little bit more louder. It's a little, yeah. Can you speak a little bit? I hear it all muddled up. Yeah.
Yeah, so say the ligand is distributed evenly across the embryo. You can have localized receptor expression and still.
Okay, localized signaling is what I would say then. It
Yes,
doesn't
yes.
matter. So it can be, you're talking about the localized receptors, maybe for the torso kinases, right?
Yes.
Yeah, okay, great. So that example, but we can probably say it is localized signaling because for the other one, for the dorsal ventral axis, it is ligand is presented only at a specific region, whereas the receptor is present everywhere.
Yeah.
Correct? Yeah. So
yeah.
that brings about localized signaling, which will start off localized gene expression as well. Very nice. Very nice. Very nice. Then anything else? I missed this
A
one.
variation in the potency of the cells.
Variation in the potency of the cells.
As they become more specialized.
Yeah. Yeah, sure. So I'm talking about all the events that have been laid out right now, the morphogenetic gradients, transcription factors, enhancer switching, RNA localization. These are all ways to create differential cell fates, and the potency will change. Yes, that's true. But any mechanistic process that will result in change in potency is what I want to understand. So anything else? Yeah, there was this cytoskeletal changes. Yeah, cytoskeletal was one more thing that you guys mentioned. Everything to do with gene expression is done. We have different enhancers that are utilized. We have different RNAs that are localized here and there.
Different types of cell-cell interactions.
Yes. Can you elaborate on that part? I mean, as in mechanistically, what process are you implying?
It could be a reduction in the cell-cell interaction when cells have to move around in the embryo to form structures.
Okay.
And in the final embryo, you will have different types of interactions in different parts of the embryo.
So essentially boiling down to the core process, which is extracellular matrix and cytoskeletal remodeling. Correct?
Yes.
That would facilitate such interactions.
Yes.
Yes, yes. So ECM remodeling. Yeah. Okay. Anything else? We haven't I mean, we don't read much about energetics, but energy, localized generation of ATP or mitochondria or metabolism is going to be also another thing that will set the ball running. I want to show one. Wait, just a minute. I want to on this note. Wait, I just want to share a paper with you guys. I don't know if you have read it, but it's a very nice paper. Yeah. So I will share this paper with all of you. Please go over it and read it because it's a fascinating paper. Okay, I'll stop share and share screen. This one. Does everyone see this paper, the graphical abstract?
Yes.
Okay, great. So since you're mentioning all these points, I think we've fairly covered all the points. We have covered all the points that you have mentioned. All of you have mentioned morphogenetic gradients, transcription factor hierarchy, enhancer switching, RNA localization, and that is different. Then localized translation. Then translation repression is the other one. So if we talk about localized translation, we are also I think I also imply translational repression. Like bicoid will repress the translation of caudal in the anterior end, and caudal will do it for bicoid on the posterior end. Then localized signaling because of localized expression of receptors or ligands. And then the other thing is cytoskeletal remodeling, either by facilitating, either in terms of actin microtubule dynamics or ECM remodeling, which will be necessary for cells to attach and detach as patterning happens. Okay, so this part is, this is just a paper that talks about energetics, and I think I will not go over the paper, but I'll just briefly tell you what the paper talks about, because why metabolism is going to be also a key driving factor for early development. And this paper does it with some level of granularity, which is very, very neat. How many of you read this paper? This talks about, how many of you have read this paper? This is about nuclear localization of the mitochondrial TCA in the mammalian embryo. Has anybody read this manuscript? Everybody can see the slide, right?
Yes.
Okay, but you guys have not read this paper, right? Or have come across this paper before?
No, not me.
Okay, so I'm assuming, since the silence is, I will assume that not many. So this is actually a very nice paper which talks about. So we generally see now metabolism as a means to provide energy and ATP, and if the cell is low on ATPs, then of course the cell undergoes an arrest or is unable to function because it is deprived of ATP. Now that has been our whole perspective on metabolism, and mitochondrial enzymes are always restricted to their expression in the mitochondria, and that is where they are going to be central for their function. And if the mitochondrial enzymes are to be localized elsewhere, if they are present outside the mitochondria, can do other things apart from generating ATP for the mitochondria, have not been asked, have probably been asked, but or never thoughtfully done or never explored, because the idea that mitochondrial enzymes are going to be restricted to the mitochondria and important for mitochondrial function and ATP generation is the idea that we have, that we all carry. Now, this paper takes early pre-implantation mouse and human embryos and shows that a part of the TCA cycle runs in the nucleus. So the enzymes that are normally seen in the mitochondrion are localized into the nucleus. And in the nucleus, they drive a part of the TCA cycle. And the metabolites generated by this part of the TCA cycle are essential for the activation of the zygotic genome. This is essentially what the paper talks about, and this is essentially what is shown in the graphical abstract. So now the paper starts exploring the whole idea of mitochondrial enzymes and relocalization, and how they discovered this thing. But we have seen the same thing in Drosophila embryos, that prior to the zygotic genome activation, the maternal components are what drives the early rounds of proliferation and function establishment of the gradients. Following that, the zygotic genome activation happens. And all the points that we talked about are transcription factors, correct? Now, imagine how transcription factors actually work. Now, transcription factors will require the genome to be remodeled. You need to activate the genome. Now, how does the genome get activated? Even if transcription factors are present, the activation of the zygotic genome will require acetylation, methylation, or you will need to activate some genes, you will need to repress certain genes. Now, the transcription factors can only work with a substrate that will activate the genome or inactivate the genome. And these substrates are essentially metabolites themselves. So you have to activate a certain gene, you want to acetylate it. Now, where is acetyl going to come? From acetyl-CoA. Now, the question is, okay, fine. If acetyl-CoA is needed for zygotic genome activation, it relies on the mitochondrion to derive it. Okay, so which means that the mitochondria has to be very busy and active. But what started off with this in this paper is when they actually, I will strongly suggest everyone to read this manuscript. So what this paper talks about is how when they started growing embryos, these early pre-implantation mouse embryos, and deprived them of certain, in the culture medium, deprived them of certain metabolites. So if the mitochondria is active, deprivation of these metabolites wouldn't lead to any kind of arrest or any kind of developmental delays. But what they observed is that certain metabolites were very key in the development of these early pre-implantation embryos. And gradually, the paper goes about seeing the mitochondrial activity and talks about how the mitochondria is inactive and not functioning. So where is all the acetyl-CoA coming from, and how is that the metabolites that in the culture medium, when they're missing, they result in a block. So it turns out that most of the mitochondrial enzymes, like aconitase, isocitrate dehydrogenase, are present in the nucleus. So this is the cytoplasm. And the remarkable thing is that there is temporal localization of these enzymes in the nucleus. They're present prior to zygotic genome activation. And post-zygotic genome, they are no longer seen in the nucleus. So what they see, when these are all pyruvate dehydrogenase, these are all present in the nucleus and not in the cytosol, and not in the cytoplasm. This is a two-cell staged mouse embryo. It has undergone the first division. And at the two-cell stage is when, and at the four-cell stage, 2 to 4, is where the zygotic genome activation happens in the mouse. So all these enzymes are present in the mitochondria, and what this paper shows is that these enzymes, all the way from pyruvate dehydrogenase to isocitrate dehydrogenase, they're all expressed in the nucleus, and that is localized, this localized generation of acetyl-CoA from these steps of the part of the steps of the TCA cycle is what leads to the development of the switch of the zygotic genome. Now, with this example, I think one should also try and ask, how does localized metabolism control other aspects of development. This paper does it in mouse and human embryos. I think the last figure is, this is quite a remarkable discovery, I would say, because we have always seen TC enzymes localized into the mitochondria. Now, this is, I think, a human embryo. And in human embryo, and what is unique is that the timing of localization of these TC enzymes in the nucleus is reliant on when the zygotic genome is going to be activated. Now, in the mouse, the zygotic genome activation happens between 2 and 4 cell stage. At the 2 cell stage, the zygotic genome needs to be activated. So all the enzymes that of the one part of the TCA cycle are present in the nucleus only at the 2 cell stage, and afterwards they are no longer seen. Now, in the human embryos, what was seen is that in the human embryos, the zygotic genome activation, so these are 2 cell human embryos, no localization of PDH, which is pyruvate dehydrogenase. But at the 4 cell stage, they start seeing localization of this enzyme in the nucleus. And between 4 and 8, the zygotic genome activation has happened, after which the nucleus is again derived of the mitochondrial, of the pyruvate dehydrogenase enzyme and more localized into the mitochondria. So, and restricted only into the mitochondria. So what this means is that, again, the same thing like the RNA component that we see that they're specifically localized at specific locations. These enzymes, their localization into the nucleus is timed with the zygotic genome activation. In mouse, they're only seen at a 2 cell stage between 2 and 4. And afterwards, they're not seen. And in the human embryo, beautiful again, the enzymes come into the nucleus. When the zygotic genome has to be activated, they function, they derive acetyl-CoA, activate the genome, and then the enzymes are out of the nucleus back into the mitochondria. Not that they are going back into the mitochondria, but I think what what they hypothesize is that it's not that the mitochondrial enzymes move into the nucleus, but that when these proteins are being localized into the mitochondria or elsewhere, the localization of these enzymes is directed into the nucleus. So however the regulation of this happens, this is still unclear, but it is the localization, instead of going into the mitochondria, some of these enzymes end up into the nucleus. Yeah, so the one point, apart from all the transcription, translation, and cytoskeletal remodeling, the one point that is kind of very prematurely understood is localized metabolism that is going to support development. And now, post this paper, the expression of these mitochondrial enzymes have been detected also in cancer cells. So cancer cells seem to be utilizing the same machinery, the same approach to proliferate and to model their genome to be causing, I think in prostate cancer cell lines, and also in oral carcinomas, the expression of the TC enzymes are seen in the nucleus. Yeah, so I will share this paper for your general reading, and it's a new aspect, and it's a very nice, simple paper to read, but a very exciting read. Okay, any questions on or any thoughts on this this idea from anyone?
Hi, Tina, this is Utkarsh here.
Hi, Utkarsh. Oh, absolutely fit for you, huh?
Yeah, yeah. So you said that, I mean, they're driving the TCA cycle in the nucleus
Yes.
to basically provide the acetyl-CoA, right?
Not the entire steps of the TCA, but you see
Yeah,
the pyruvate dehydrogenase all the way to IDH.
a part of it. Yeah. So if you were to, let's say, block the TCA cycle and just provide acetyl-CoA,
Yeah, they can.
so
They've
they
run,
would you see the same thing then?
yeah. They've done those experiments. You read this.
I see. Okay.
Yeah. So in fact, they have actually um, mm, they've done some labeling experiments, either in this paper or I just know that they've done labeling experiments to show that that was one of the reviewers' experiments they had to do.
I see.
Yeah. They gave labeled something, and however they measured it, yeah, they are able to rescue.
I see.
Mhm. I mean, read
Okay.
this paper. I forget details about it, but they've done that experiment.
Okay.
I think in figure 6, a part of figure 6, and their supplementary figures will tell you as well.
Mhm.
Yeah.
Okay, and just one more thing. So, like, what kind of metabolic profile will a cell attain must really, I mean, should also be dependent on the vascular development, right? Like accessibility, which will dictate in some sense the accessibility to nutrients. So in that sense, like how does vascular development happen in Drosophila? It
So
is
in Drosophila, so right, I mean, in Drosophila, you mean, right?
yeah.
Or not? No, are you talking about pre-implantation mammalian embryos or?
No. In Drosophila. So this is about the segmentation. Till the
Okay, because
segmentation.
in pre, just let me segregate the thought here a little bit as well. So in pre-implantation embryos also, vascularization has not kicked in. So at this point of
Yes.
time, the embryos are just have not been embedded. So everything so there is no essential vascularization that feeds into these embryos. They are living in the fluid around, and whatever they um, so there is no active process which is delivering nutrients into these pre-implantation embryos. Post implantation, vascularization will be so till then this embryo is actually a very autonomous unit, and whatever it takes up from the what do you call the I mean, whatever it takes up as metabolites, but essentially till this point of time, what it generates by itself is going to be an autonomous. There is not much interaction or connection with the extracellular environment, active process. Now, in Drosophila, vascularization will be through the trachea. That, I mean, the vascular component doesn't exist in Drosophila. So
It's essentially diffusion sort of.
yes, diffusion. The only thing that is comparable to vascular network is the trachea. Now, trachea is utilized to transport oxygen, and the tracheal innervations are seen in. So during, okay, now I'll stop sharing this slide. Just let me. Okay. Does everybody see this one? Slide back to gastrulation.
Yeah.
Yeah. So at this point of time, you see these small pits that are forming here.
Yep.
These are the tracheal pits. Now, at this in this stage of embryonic development, embryonic development, the trachea is still forming. It has not vascularized extensively to be able to support oxygen delivery or anything. It here as well, the embryo is a very autonomous unit and a very closed system. The trachea is not active. It is building its vascularization or building its network. Now, once the embryo has emerged as a first insta larvae, what happens is that there is an oxygen bubble that will move all along the, I mean, I don't have a trachea slide here. No, I don't have a tracheal slide, unfortunately. So an oxygen bubble moves along the, so these two ends that you see here are actually the opening of the trachea, and all the way until embryo, they are closed. Now, once the embryo hatches as a first insta larvae, the tracheal pits here, the tracheal openings will open up, and an oxygen bubble will move all along the trachea and its branches, and this will result in the opening of the trachea. Now the larvae is going to be actually utilizing trachea for supporting oxygen delivery. Apart from that, everything else is through diffusion. So trachea doesn't support any kind of nutrient transport. The only transport that it does is oxygen, but oxygen also can freely diffuse through the cuticle. So it's an open circulatory system in Drosophila, and it doesn't rely on the, so even if the animal is, even if the animal doesn't have well-formed trachea, it's not that the animal is going to not see oxygen transport, because oxygen can freely diffuse. But it is just to support better oxygen delivery into the deeper tissues, like the wing disc or the imaginal disc, or I mean, now there are more functions of the trachea. What happened? Sorry, my Photoshop. No, I don't know what happened. Okay. Yeah. So that is how the vascularization is seen in Drosophila. It's not as extensive as in mammals. I mean, it's not at all extensive. Does that address your point?
Yeah. So essentially, before that, is the embryo growing in some sense a hypoxic environment?
Yes. 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 The embryo is quite hypoxic.
So HIF would be
not
like,
because. Yes.
so have people done
HIF is
some kind
expressed.
of like a HIF inhibition experiments to see like how development happens in the absence of HIF transport to the nucleus?
So not that people have done, but essentially now we're asking those questions for a different reason. But HIF is expressed in very early embryos, and HIF is going to be essential for the early embryonic metabolic state. And I mean, it's not very oxidative, it's very glycolytic. And the expression of HIF is essentially seen all throughout embryogenesis, but detailed understand. And if you take HIF mutant embryos, they're developed, they cannot proliferate. The early syncytium blastoderm to cellularization, those steps are delayed. So that is the only understanding that we have of HIF function in early development, but not more than that.
I see. Okay.
Yeah. Any more questions? Okay, great. So I will take the liberty of explaining at least this one particular tissue that we work on in the lab, and to give you a flavor of how development will take forward organogenesis. Now, as I mentioned in our previous class, that in the process of gastrulation, you will have the mesoderm and the 3 germ layers separate out. Now, blood is a mesodermal tissue, and across systems, not just in Drosophila, but blood is derived from the mesodermal tissue. And now you have, as the ventral furrow, you have the ventral furrow which invaginates, and the 3 germ layers separate, and you have the cells in the anterior region. So you'll have a head mesoderm, and in the posterior region, you will have the trunk mesoderm. So the mesoderm across the anterior posterior axis will be also defined differently. So it's going to be head mesoderm and the trunk mesoderm. And blood in Drosophila is going to be derived from a few subset of cells that are set aside from the head mesoderm by, again, going back to the expression of some transcription factors. We call them lozenge and GCM. It doesn't matter what they are called in Drosophila, but essentially these are runt domain transcription factors. And runt domain transcription factors are seen across systems, model systems. And the specification of the head mesoderm that is going to give rise to blood cells is initiated by the expression of both GCM and lozenge. And while they are going to give rise to cells of glia, and so these head mesoderm cells, which are set aside, they will give rise to glial cells and also give rise to prohemocytes. They're also, we call blood cells in Drosophila hemocytes. So these are called procephalic prohemocytes because they're originating from from a subset of cells in the head region, which there is a common ancestor. This is a common ancestor here, which is either going to give rise to a glial cell or a blood cell. Now, this event in the head mesoderm establishes the initiation of first, and this is one of the most primitive, not primitive, one of the most early tissues to be defined before any other tissue is defined. So this happens very early stage 7 post gastrulation, one of the first events that happen before the other stages or before other steps of organogenesis are initiated. The head mesoderm is defined, and from the head mesoderm, we have a few subset of cells that are segregated because of the expression of lozenge, and they are going to be the prohemocytes. Now, this head mesoderm region is also going to give rise to cells that are of glial cells in the embryo. Okay, now these immune cells that we see here, so I don't know how many of you are aware of how hematopoiesis happens in general. So hematopoiesis in general sees 2 waves. There is a primary wave and there is a definitive wave. Now the primary wave happens in the in vertebrates. It happens in the yolk sac, and this is a very primitive. This is a very even before you have the development of the bone marrow or hematopoietic stem cells are defined. This is way earlier than that. This is within the yolk sac. There is a region called hemangioblasts, because these hemangioblasts are nothing but essentially precursors or primitive, they're precursors which are going to give rise to the embryonic immune cells. And at this point of time, there is no vascularization, nothing much has stepped in. the hemangioblast, there is a hemogenic, so the endothelium that can give rise to blood cells is called a hemogenic endothelium. And in the hemogenic endothelium, there are these precursors called hemangioblast, from where these primitive blood cells are going to be arising from. Now, this primitive hematopoiesis is then going to be later, as the embryo is developed, in a few days, transition into a second wave of hematopoiesis that is seen in the embryo, which is going to give rise to the hematopoietic stem cells in the AGM. It doesn't matter where it happens, AGM or wherever. Now, so essentially, the primitive hematopoiesis is going to give rise to immune cells that are very embryonic-like and not, and they have a certain potential. There are differences between the immune cells that come from this primitive wave as opposed to immune cells that come from the definitive wave. Now, the same thing happens here as well. So there is the primitive wave of hematopoiesis that happens in the head mesoderm, which will give rise to cells that are going to be mostly circulating cells. So this is exactly what happens in the mammalian context as well. Now, as the embryo transitions into the larvae, these immune cells that have come from the head mesoderm are going to be proliferating and seen as circulating immune cells. Now, if you can imagine, these cells in circulation are all coming, whether they proliferate and how many cells they are, they have this one origin from the head that they have arrived from, where they've been derived from. Now, in the embryonic stages as well, the specification of an organ called a lymph gland happens, which is the definitive wave of hematopoiesis. Now, in mammalians, this happens in this region called AGM. This is called aorta gonadomesonephros. So this is a common, from here, you'll have the heart origin, the gonads, and the kidney. So this AGM is a common denominator for giving rise to the heart, the gonads, and the kidney. And also from here, once the heart has been defined, that aorta region is from where the hematopoietic stem cells are going to be derived from. So this AGM is very critical for the second wave of hematopoiesis to happen, which is called the definitive wave. Now, a similar ancestor also exists in Drosophila, where an AGM-like population is seen. And from the AGM-like population is where the specification of the first cell that is going to give rise to blood precursors happen. Now that there are, in the embryo, it will be a few cells that will be the precursors to the blood cells. In the larvae is where these precursors start to proliferate. Now, this green thing that you see here running all along from the anterior to the posterior is nothing but the heart. Now, the AGM that I just talked about is going to give rise to three things. It is going to give rise to these blood cells of the lymph gland. We call the gland that holds the blood cells together, we call it the lymph gland. It's nothing to do with the lymph nodes or the lymphatics. It is just that the organ is called a lymph gland. Now you have all the three cells of the AGM here. You have the dorsal vessel, which is called the heart, which is the heart in Drosophila. It's a tubular heart. You have the blood cells, and these golden yellow colored cells are nothing but the pericardial cells or the nephrocytes, which are like the cells of the kidney in Drosophila, akin to the cells of the kidney in Drosophila. So the AGM in Drosophila as well is going to give rise to these three things: the aorta, and not the gonads here, but aorta and the mesonephros, which is the kidney, and the blood. Now, the blood cells in this very early embryonic stage are all progenitors, or all, we don't call them hematopoietic stem cells, because to prove them as stem cells, one has to do transplantation experiments and many other things. But regardless, we call them multipotent progenitors. And these cells of the lymph gland during this, so this gland is also developing in the larval stages. So from the early to a late larval life, now Drosophila larval life has the first insta, second insta, third insta, and wandering third, and a pupal stage, following which the pupa will eclose and you will an adult fly. The lymph gland initially grows and proliferates, where all these prohemocytes or these progenitors or stem-like progenitor cells are actively proliferating. By a late larval life, by a late in a third insta larvae, these prohemocytes have all proliferated and they will start differentiating. So these stem cells, these stem-like progenitor cells are not going to differentiate, but ultimately what happens is that this gland will have a lot of blood cells, but there are cells in circulation which are the cells of the embryonic origin. Now, these cells in circulation are essentially similar to the tissue. They can actually give rise to tissue-resident macrophages, the ones that are commonly in the earlier stages of vertebrate mammalian hematopoiesis. You have all the embryonic hemocytes. They give rise to the tissue-resident macrophages, which has gained a lot of popularity right now. Is essentially also true for Drosophila, that all these circulating immune cells that you see here, they also attach to different tissues, and they reside in those different tissues, like the imaginal discs and the muscle, the trachea. So they are found localized within various tissues and are supporting the development. I mean, it's not very well understood what they do in all tissues, but in some of the tissues, it has been identified that the signals coming from these hemocytes are necessary for regeneration and development. Anyways, now this definitive pool of immune cells that are seen in the lymph gland do not contribute to any immune cells in circulation at this point of time. It's like the it's very localized and very segregated pool of blood cells. Now, only in the adult or in the pupal stage, very late pupal stage, the lymph gland actually dissolves, in the sense it disintegrates and releases all immune cells into circulation. Essentially, the whole structure, even the heart is remodeled and the nephrocytes are also remodeled. So a fly is born twice, right? So once as a larvae and then as an adult. So at the pupal stage, it will again undergo a lot of morphogenesis, remodeling, and development is going to shape up here, which will give rise to an adult fly. Now, at this point of time, the blood cells of the in the pupa are both the circulating ones that are going, now since essentially everything is in circulation, now all the immune cells in pupa and the adult are going to be seen from the lymph gland, from the definitive wave, and from the early embryonic wave. Now, what does the lymph gland look like? This is a lymph gland, a third insta lymph gland. The green cells that you see here are all the progenitor cells that I mentioned them as stem-like multipotent blood progenitor cells. The red cells that you see here are all the differentiating blood cells. Now, this is a model that we use in Drosophila to understand hematopoiesis. And we talk about these cells as stem-like cells because these blue cells that you see here are all niche cells. They're cells of the niche because signals, we know that, and a common idea that connects all stem cells together is that they're dependent on signals coming from the niche. So the development of these cells, of the progenitor and maintenance of these cells as progenitors is reliant on signals coming from the niche. So the PSC, it's called the posterior signaling center, releases signals that are sensed by the cells of the the cells of the medullary zone. So these are the medullary zone cells. We call this as a medullary zone, but essentially these blood progenitors, which are stem-like, they need the signals coming from the niche for their maintenance in an undifferentiated state. Now, as they move again, so if you see the signal from the niche, it has a gradient-like phenotype. I will show you in a bit if it is here somewhere. Sorry. No, not here.
Tina, I had a question.
Yes.
So just wanted to ask if there's when in the adult stage it disintegrates, the lymph gland disintegrates and the immune cells are distributed all over. Are there any mammalian counterpart of microglia in Drosophila brain as well?
See,
Like,
correct. Drosophila doesn't have microglia kind of a thing, but they have glial cells that do microglial-like function. Now, I mean, the question that you ask is not very well explored because what we know is that these immune cells, unlike in the vertebrates, it's now being explored. There is glia in the brain of the Drosophila embryo, glia in the brain of the Drosophila embryo, larvae and adult. But if the immune cells are also contributing to the glial compartment in the adult, not being explored. Now, in the embryo, there are separate glial cells. But what is being reported is that these macrophages, essentially what the immune cells in Drosophila are all myeloid. There is no T and B cell. So there is no lymphoid arm here. Now in these early stages, the macrophages here support the kind of function that the microglia do. So they're essential for pruning, they're essential for fine patterning, but the nervous system is also developing at this point of time. And the kind of functions that these macrophages contribute, so these macrophages are not just forming, they're also functioning in early embryonic development. And what has been noticed, what has been reported, is that these macrophages perform functions like what microglia can do. So neuronal pruning, proliferation of the neuroblasts, and such functions are controlled by these macrophages. Now, how they control remains to be understood in detail, because I'm citing very old literature right now here, but the understanding of the functions by these macrophages was mostly signaling driven. So they release some signal which is necessary, either ligands or some JAK-STAT pathway ligand or some ligand, or activation of a signaling cascade through which the central brain patterning is achieved. But these are macrophages, right? So they have the capacity to scavenge, and they
yes, sorry.
Yeah, but any aspect of their scavenging function on central brain patterning, or like the function that microglia can do, has not been explored.
Okay.
Across development from embryo to adult.
Okay. But these macrophages are also found in the Drosophila CNS.
So they do not, so now at this point of time in the embryo, they have not, they're in the milieu where everything is quite open and accessible. So the
Okay.
system is not yet closed.
Okay.
They don't really migrate into the brain and give rise to the glia in the brain, if that is what you're asking.
Oh, okay. No,
So
what I was thinking is irrespective of
they
glia, but
localize
they,
to the brain
yeah.
and then get trapped. No.
Yeah. Okay.
But what I have seen is that they can attach. So when there is an active inflammation response in Drosophila, they can line up all along the brain. They do not invade or they are not invasively going into the brain, but they can attach onto the cortical glia. So the glia that covers the brain is called cortical glia, and these macrophages essentially can line up all along the central brain. Now, it has not been explored if they're going to be communicating with the cortical glia or then transition into cortical glia. These are all open questions that have not been asked.
Okay.
Yeah.
Thank you.
Yeah, sure. So I mentioned that these progenitors are stem-like, dependent on signals coming from the niche, and the immune cells in Drosophila are mostly myeloid-like. And what are the cells that are seen in Drosophila then? The cells that are seen in Drosophila are 95% of them are plasmadocytes. Plasmadocytes are nothing but macrophages. They have exactly the same properties as macrophages. They are very actively scavenging, foraging, necessary for tissue remodeling, cleaning up debris, and immune functions, also eating pathogens. And the remaining, in homeostasis, the remaining 5%, so 95% are these, the remaining 5% are these crystal cells. So essentially these are the platelet-like crystal cells. So if any wounding happens, these cells will function like what platelets do, erupt, they will release these crystalline inclusions that will help in wound healing and blood clotting and close the wound. So in homeostasis, one sees, so all the red cells that you see here are essentially all the plasmadocytes lined up along the outer surface of the gland. And in between them will be a few crystal cells. Now, not often, but there is this other third cell type which is seen called the lamellocyte. They are very large flattened shaped cells, 40 microns. So this is a plasmadocyte. This is a very large this is a very large lamellocyte. Now their formation, so these are not macrophages and neither they are crystal cells, but what these kind of lamellocytes do is like what our, we also have these large dendritic cells or parasitoid. So when there is a deposition of a parasitoid egg, which is very large, very much larger than what a plasmadocyte can engulf, these lamellocytes actually are formed and they enwrap. So this is say the wasp egg that has been deposited. So those lamellocytes which I showed you will actually cover this wasp egg like a coat and then neutralize it. They will release reactive oxygen species and then they will encapsulate the wasp egg and then result in an oxidative burst which will kill the wasp egg. So you can see here all the plasmadocytes and crystal cells. Plasmadocytes will be, so there is a breach of the epithelium. The plasmadocytes will come to the spot, neutralize, they will scavenge the phagocytose, the bacteria or the debris, clear off. Crystal cells will come and the crystal cells will release the crystalline inclusions, formation of the heart clot, which will result in the closing of the wound. So essentially this is a wound healing response. Now the wasp egg response that I just mentioned. So this is a wasp which is injecting, so when it infects, it has an ovipositor here. And through the ovipositor, it deposits these wasp eggs, this wasp egg, and they're very large. They're very large wasp eggs. Now, once the lamellocytes enwrap the wasp egg, they form these black melanotic capsules. And they are black because they've been melanized and killed. So these are the killed wasp eggs. And these are the lamellocytes that form, which will enwrap the wasp egg and result in its killing. Now, now, what I want to bring about here is kind of a similarity between the development of blood in vertebrates. So gradually you can see there is stepwise loss of self-renewal. So the long-term HSCs which are seen in the vertebrates, they have this long-term renewal capacity. They, meaning that they're really stable stem cells, and they self-renew, they proliferate to give rise to differentiated cell types, but they also self-renew themselves. So they're not lost with time very easily. So they're called the long-term HSCs, and they give rise to the myeloid progenitor cells from where all the myeloid cell types are going to be originating. And in vertebrates, there is this huge plethora of markers that are utilized to identify each subtype. In Drosophila blood development as well, there is this hematopoietic stem or progenitor cell which self-renews itself, and it gives rise to a common myeloid precursor. So this is Drosophila, I'm sorry. So this is a Drosophila stem-like precursor cell, and it will proliferate to self-renew and give rise to all the three different lineages that I mentioned: the plasmadocyte, lamellocyte, and crystal cells. And the mechanism or the pathways that are involved in these processes are kind of similar. So all the GATA transcription factors are necessary for self-renewal. In the vertebrates, serpin is a GATA family of transcription factors. Serpin belongs to the GATA family of transcription factors, and the prohemocytes here in Drosophila utilize. Essentially, what this slide is telling you is that the mechanisms set in place in Drosophila hematopoiesis are quite parallel to the processes set in place even in vertebrates. And again, the idea of transcriptional hierarchy. In regulation, you have a prohemocyte and the transcriptional regulation by activating different target genes and different signaling pathways, which will result in different cell fates. So like one person mentioned, potency. So the potency of a prohemocyte changes to be more specialized by utilization of the different signaling pathways. If they utilize GCM, it becomes a plasmadocyte. If the cell sees more JAK-STAT or sterate NOT signaling, it forms a lamellocyte. Whereas if it sees only very high NOT signaling, it will form a crystal cell. So depending on the type of signal that you see, you can actually, the cell decides what its fate is going to be. Now, essentially after this, essentially after this, everything else is signaling, and I can tell you what the blood system is, utilizing different signals for, but one of the aspects, so while a lot we understand about stem cells or development is through niche and niche dependent signals, one of the things that I think has picked up recent popularity is in the field of stem cell maintenance is non-autonomous cues, meaning niche independent. So if you understand how hematopoietic stem cells are, now the unique property, I'll just go back to one slide, which I just skipped, sorry. Yeah, if you go back to the hematopoietic stem cells in vertebrates, they have a very unique character. The unique character is that hematopoietic stem cells from the bone marrow, leave the bone marrow, and they migrate and then they home back into the bone marrow. But while they're migrating, they don't lose their property of being a stem cell. But an important property of the hematopoietic stem cell when it migrates is also it goes back and homes into the bone marrow, hematopoietic niche. So the question then is, once it's outside the niche, then it's no longer seeing a niche signal. Then how does it maintain itself as a hematopoietic stem cell? So this is quite a property of hematopoietic stem cells, that they can leave the niche and roam around and then go back and home back into the niche. So the idea that niche is necessary is fine, but niche is not perhaps the only thing and sufficient, because there has to be niche independent cues under these conditions that keep a surveilling hematopoietic stem cell still a stem cell. Now, if you see the hematopoietic niche, even in vertebrates, has very different types of cells. Now, in addition to the endothelial cells and the sinusoids where the hematopoietic stem cells reside, there are the glial cells and the nervous system, which invades into the bone marrow. Now, there is not much information, but there are a few papers that talk about how the sympathetic nervous system or the glial cell signaling is necessary for maintaining stemness in hematopoietic stem cells of the vertebrate niche. Now, in Drosophila, as you now understand that the lymph gland is like freely exposed there's no bone marrow in which it resides. The hematopoietic stem cells the hematopoietic progenitors are there in this lymph gland, which is accessible, which is in the open system and very accessible to the circulating blood. So there is no vascularization, so everything is an open system here. Now, what has been observed is that the progenitors of the blood, even in the lymph gland of Drosophila, are reliant on cues coming from the brain. So the central brain, and now there is no nervous system that invades the blood system, but what the nervous system here does is that it signals through the release of secreted factors, which are which are present in the circulating hemolymph, and then by the blood progenitors of the lymph gland detected, and post detection they run a cascade of events that are necessary for development, maintenance, differentiation, whatever. So one of the signals that has been identified is GABA, which is a neurotransmitter, but here GABA is released from the brain, sensed by the blood progenitor cells, and necessary for their maintenance. Now, I'm sure there'll be many other cues that the brain signals, but that is something that is still people are still working on it. And now I can leave you to imagine how, now it is all about what blood cells can do and what blood cells are. But this slide here essentially summarizes what I just mentioned in my, you can read more about blood development and the utilization of Drosophila to understand blood-related disorders. That's not what I want to dwell on, but that the idea that Drosophila development is comparable, Drosophila hematopoiesis is comparable to hematopoiesis seen in vertebrates. You have the head mesoderm. From here, the first cells of the blood are going to originate, first blood cells are going to originate. And these embryonic blood cells are going to be circulating in the embryo and performing embryonic functions necessary for embryonic development, like what I mentioned, CNS pruning and development of the central nervous system and also of the peripheral nervous system. Apart from that, not much is understood about any other function that embryonic immune cells do, but I'm sure there must be. Now, this is the dorsal vessel. From the AGM region that I mentioned, you will have the heart and the cells of the lymph gland that are going to get specified. And this is the cross-section view. Now, in the larval stages is where this organ is going to be developing and giving rise to the lymph gland. The lymph gland is not just reliant on signals that come from the niche, but as I mentioned, it is also dependent on non-autonomous cues. One of the cues that I mentioned are ones coming from the brain, but there are other cues coming from other tissues as well. So the idea that there is a common origin of blood, both for the embryonic wave and the definitive wave, is something that makes this system a very exciting system. So if anybody wants to understand how hemangioblasts give rise to the three, how the specification of hemangioblast happens to give rise to the three different types, the heart, the blood cells, and the nephrocytes. This is a good system to ask because there is sequential segregation of three different tissues happening from a common origin. And from the angioblasts, there is the origin of blood cells. Now, the whole idea of where does the hematopoietic stem cell come from? What are the signals? When is the first hematopoietic stem cell derived from? Because essentially, if you understand this, you can actually take these hemangioblasts, grow in culture, and then derive hematopoietic stem cells out of them. So the whole idea of bone marrow is to get hematopoietic stem cells. Very hard to culture hematopoietic stem cells. So the whole idea of bone marrow transplant is to be able to have enough hematopoietic stem cells that will be giving rise to for the therapy. Now, the utilization of such model systems is useful because A, it's simple to work with, B, the system has commonalities, and the signals are not going to be very different. Yes, the complexity will be very different. I mean, the complexity is not the same, but the core components are there. You have a hemangioblast, which is going to give rise to the three types. So whatever a hemangioblast needs to give rise to a blood cell exists in this model system. And therefore, if one wants to understand what are the events or the sequences of events that lead to the formation of the first hematopoietic stem cell, Drosophila can be a very useful model system to ask that question. Not that anybody does that, is doing that in the field that I'm aware of, but yes. So this is the value of using a simpler model system, because many complex processes are relatively simply presented in such model systems. So instead of approaching a very complex problem in a complex model system, you ask a complex question in a simpler model system, address it, understand, and then apply those ideas to a more complex system and see how much of it holds true. Now, Drosophila has been very fantastic with that regard, because it has been fairly consistent, fairly conserved across model systems. So yeah, with this, I will end my class here. Anybody has any questions for me, I will be happy to take. But after this, I'm done with my class for this semester. And anybody has any questions can ask me now or can email me later as well. Any questions? Okay, so then thanks for attending the lecture, and I hope I will go over the assignments, and I will get back to you with my comments in each assignment as in the comment section. And if there are any doubts as you go about reading anything pertaining to what we have covered in the course, do not hesitate to drop in an email, and yeah, I'll try to explain I will try to go over it and see how we can simplify it. Yeah. Okay, great. Then thank you guys.
Thank you.
Thank you.
Thank you.
Thank you, Tina.
Thank you.
Thank you.
Sure, thank you. Hi, Varsha.
Yes, Tina, hi.
So I'll send you these slides and the paper that I want to read. I mean, there's nothing giving them as an assignment, but just for
Okay.
reading, I'll forward the slides and
Okay.
yeah. And now
Yeah, sure.
the assignments with me in the Moodle, right?
Yeah, the assignments are all in the Moodle. So should I download them and send it to you, or?
Do that. That will help me a lot.
Okay, I'll do that. I'll send in the other assignments also, along with the ones and.
Yes, great. That will be
I'll mail, yes. I'll mail them to you by today afternoon. Because, yeah.
good. So after this, I had only one class, or I had more classes?
No, you only had two Wednesday's class. So there is no class on Wednesday.
There's no class on Wednesday, and I'm done with my classes.
Okay, Tina. I'll write them a message saying there is no class on Wednesday and Friday. And yeah.
Who's next on?
Raj is teaching next.
Okay, great. Okay, fine. Thanks, yeah. Thanks so much, Varsha.